Now, students, we are going to study enzymes in our biochemistry class. You have already studied enzymes in your matriculation in biology. However, in today's lecture and in our other lectures, we will discuss these enzymes in detail. In this lecture, we will study that what is the nature of enzymes, how you can define enzymes, and what is the history of the enzymes. So, moving towards the introduction of enzymes. Enzymes are the biological catalyst. They speed up the reaction. Most of the enzymes are the three-dimensional globular proteins, that is, they are tertiary or quaternary structures. We have discussed these proteins in our previous video lecture. Now, some special RNA species also act as enzymes, and we call these special RNA species as ribozymes. Example is hammerhead ribozyme. So in this slide, I have discussed 3 points. First one is that enzymes are the biological catalyst. Secondly, most of them are the three-dimensional proteins. Thirdly, some special RNA species also act as the enzymes. Now, what is the definition of enzymes? Enzymes are the proteins or RNA which are produced by the living cells and are highly specific and are highly catalytic to the substrate. So these are the macromolecular biological catalysts, also known as the biocatalyst. Now, enzymes act on the molecules, and these molecules are called the substrates. Now, after acting on these substrates, these enzymes convert these substrates into different molecules, and we call these different molecules as products. Now, what are the examples of the enzymes? Lactases, alcohol dehydrogenase, DNA polymerase. Now, what are isozymes? Different enzymes that catalyze the chemical reaction, that basically catalyze the same chemical reaction, are called the isozymes. Now, what is the history of the enzymes? We already know that fermentation process is associated with a scientist, that is Louis Pasteur. We have studied his research in detail in your biology class in matriculation. So, in 1850s, there was a scientist, Louis Pasteur. He concluded that fermentation of sugar into alcohol by yeast is actually catalyzed by ferments. But he postulated that these ferments are actually inseparable from the yeast cells. And this concept, that is, the ferments are inseparable from the yeast cell, is called the vitalism. However, in 1897, another scientist, that is Edward, he discovered that definitely yeast extracts could ferment sugar into alcohol. But he concluded that this fermentation process is promoted by the molecules that continue to function when removed from the cell. So basically, he was trying to say that these ferments are actually inseparable, not inseparable, but these are separable from the yeast cells. However, another scientist, Friedrich, who actually used the term enzymes first time for these molecules. He called actually these molecules are the enzymes. So this is the picture of Friedrich, the scientist who firstly used the term enzymes. However, in 1926, there was isolation and crystallization of the enzyme, that is urease by the scientist James. So urease was studied in detail by James, and he worked very much on that enzyme. There he postulated that after studying the urease, he postulated that enzymes are actually the proteins. Then in 1930s, we have other scientists, that is Sumner. He also worked very much on enzymes after John and Moses. John and Moses actually catalyzed pepsin and crystallized pepsin and trypsin. They worked on purification of pepsin and trypsin. So different scientists came and they worked on the enzymes. As we were moving in advanced stages, as we were moving, there was working on the purification of the enzymes. Then came the scientist Haldane. He wrote a thesis on enzymes. He also gave different theories about the enzymes. Then later, different scientists came. They again worked on purification of the enzymes. They dated the structure of the enzymes, they understood that how the enzymes work. So different scientists came, they discovered different enzymes. They also studied the structure of the enzymes. So this was all about the history of the enzymes.